Mr. Chairman, ladies and gentlemen, I apologize for speaking English, but I will speak slowly. It's a pleasure for me to be back here in Shanghai, and it's also a pleasure to talk about what I think is going to be the most important development in medicine in the future. We have seen over the last few years the Human Genome Program, whereby the genetics of each of us can be determined completely. All of us are governed by our genes, and our responses to drugs and to surgery are dependent upon each of our personal genes. So I think in future, even in the field of ophthalmology, before we operate on a patient, we will need to know something of their genetics, because that will determine their response to what we're going to do. I have a particular interest because for many years I was co-chairman of the Retinitis Pigmentosa Foundation, and I also had the first patents for refractive surgery and co-founded the first company through the FDA. So in the first place, I want to know the genetics of my retinal patients, and in the second case, I don't want to cause any harm by just doing refractive surgery. And we have done 40 million refractive surgery cases worldwide, not knowing anything about the long-term consequences. So my talk this morning is, I think, unique in that it's going to cover the first screening genetically for ophthalmology surgery patients. So, if you think about medicine, all doctors should take the Hippocratic Oath. And one element of the Hippocratic Oath is do no harm. And so that's, I think, the most important thing. Can we prevent ourselves from causing harm? Well, if we look at the cornea, we've known since the last century before last that many patients had a condition called granular dystrophy. And in these granular dystrophies, the individual had a disturbance in their cornea, which was disturbance in transparency. And then almost 100 years later, a second sort of granular dystrophy was described, predominantly from a family in Italy, Avellino. Now these granular dystrophies clinically look very similar, but slightly different. In the earliest described versions, the type 1, we have a distribution of areas of corneal opacity. In the type 2, these areas tend to be larger and more disruptive in terms of light. Now, the first scientific studies really concentrated on the histopathology of these conditions. And it was noted there was a slight difference between the type 1 and the type 2. In the type 1, the deposits were mainly hyaline deposit, whilst in the type 2, the deposits were amyloid. Although occasionally you had mixed deposits in Avellino. And we saw similar deposits in lattice dystrophy. So the earlier studies, histology, which tells us there's a slight difference in the causes of these areas of loss of transparency. But the real advances came about when we began to study the genetics. And this is a very interesting paper because it's a very simple paper to understand. But basically what the earliest genetic studies said was that these dystrophies tracked to chromosome 5 with two major areas within the chromosome causing type 1 and type 2. This is an autosomal dominant condition. So when we actually go back and look at the patients again, we see that in the homozygotes, where both chromosomes are affected, we get a very early onset, very severe form of the disease. And you can see here, this is childhood, and very early on, late teens and early 20s, a very poor cornea with terrible prognosis. By contrast, the heterozygotes may be very late onset and initially very marked. But we know that these individuals, over a number of years, will also go to a more severe area of corneal loss. So let's go back to the genetics for a moment. And now we know that within each of those areas, the two main areas, there are many different mutations, deletions, additions, and transfer of the elements of the genetic code. Just look at the pace of movement. In 2006, we thought there were 38. 2014, we have 63. This year, it's in the 70s. So this group of patients represent a huge number of genetic defects, but just in two areas of the individual chromosome. And here in China, you can see quite a number of patients show these different specific mutations, but quite large numbers of cases. This is a worldwide problem that has been very much underreported. Now we know the individual mutations, we can look at those in terms of where they occur in the cornea and the time base of that occurrence. This is the time in decades, and this is the location of these specific lesions within the two areas of the chromosome. So we're beginning to build up a knowledge base on the basis of genetics, which we can cross-correlate with our patients. Now, the most important element of all of this is the fact that all of these mutations affect TGF-beta. So if we look at the cornea, we know it's a simple 5-layered structure, but within the stroma of the cornea, we had predominantly collagen, but also fibroblasts. And these fibroblasts maintain corneal transparency. They look after the basic biology of the collagen under normal conditions. But what happens when we as surgeons interfere? Well, the first refractive surgery was PRK. And in PRK, we simultaneously damaged the epithelium and the underlying stroma. This resulted in interaction between cytokines released from the epithelium and cytokines released from the keratocytes. These two cause reactions within the keratocytes that produce atypical collagen, atypical gags, and haze. So as a result of a surgical intervention and the effect on these cellular systems, we already know that this will cause activation of TGF-beta and transient haze. By contrast, when we did LASIK, we no longer damaged the epithelium, but now we're still damaging the keratocytes, and we're still causing changes in TGF-beta. Now imagine doing any of these procedures on a heterozygote with granular dystrophy, because we know the defect in the granular dystrophies is in the TGF-beta cascade. And now we have already surgically stimulated this system. We will get activated keratocytes. Now in my group, we have developed a system whereby we can take human corneas in organ culture, and we can keep these going for 4 to 6 weeks. We can do any sort of surgery on the model, and then study the wound healing in human tissue in organ culture. And the time courses are approximately the same. When we look at 2 different forms of refractive surgery, we see with PRK and LASIK, we get, first of all, removal of the keratocytes, and then they come back in that activated state. So just think of surgery for a moment. You have a patient who appears to be normal, but in fact is a heterozygote. You carry out the surgery, and within a few months or a year or so, you have caused a huge reaction in that patient. Totally unnecessary if you had understood the genetics. I think this is really a concern now for all refractive surgeons. You take a patient that looks normal, you don't understand their genes, you do the surgery, and you cause a change in their cornea, which effectively means that you've accelerated the disease process in their eyes. Now you can say, I've been doing refractive surgery for many years and I've never seen one of these patients. When you start looking at the distribution worldwide, and this is one study from the Singapore National Eye Centre, you see there are many more patients than you would have thought. The company that is carrying out the genetic screening, Avellino, did their own study, and in fact looking at type 1s and type 2s, and those conditions where we don't know, you can see it's a problem for every country in the world. So why wouldn't all of us do a very simple test? Why wouldn't all of us take a buccal swab and screen for these mutants? This is the tip of an iceberg. You can see there that the early studies are suggesting that the incidence here is about 1 in 1,000 patients. But just think, when we can screen for keratoconus, when we can screen for all corneal dystrophies, when we can screen even for the underlying biomechanics of the cornea, when we can screen for virtually every form of genetic disease in every tissue in the eye. So I'm making a plea here today that please begin to think of genetic screening. It's inexpensive, it's very fast, and your patients will be extremely grateful. Thank you.
Thank you, John Marshall。这个给我们这个。